Good morning. So I'm Rick Asoyan. I am one of the co-directors for the Center in Engineering Mechanobiology. Welcome to everybody here. My presence up here is going to be very short today. I'm here to introduce Becky Well, who's sitting right over here. Becky and I hope to meet all of you over this next 1 to 2 weeks. Becky's lab is very interested in the mechanobiology of the liver, but particularly relevant for today is she is one of the masterminds behind the the boot camp. She is in charge of the educational component of the Center for the University of Pennsylvania, and she and Ram Dixit had organized this boot camp session. And I'm going to turn it over to Becky, who is going to give you an overview, and then we'll start the first lecture.
Okay. All right, I just want to welcome everybody. I know we have 10 graduate students here. At least 5 of you have traveled from quite far away, and I just want to welcome you. This is very exciting. NSF Science and Technology Centers are really based on the concept of graduate education. So you guys are truly the foundation of our center, and I just want to welcome you to what I hope is a really exciting boot camp. So I want to give you a little bit of an overview of what CEMB is, and then we'll go through a few details of what's going to happen today and for the rest of the week. So the overall CEMB was started. It's actually been a concept that people have been talking about for many years, at least 5 or 6 years. And the basic idea is that oftentimes biologists and physical scientists don't talk to each other and often can't talk to each other. They just don't have a common vocabulary that allows them to meet in the middle and discuss collaborative work. And the concept behind this center is that that kind of collaborative work is critically important, and that we actually can help train students as well as undergraduates, students at the high school level and their teachers, postdocs, and even faculty to be able to have these common conversations. It doesn't mean that if you're a mathematician or a chemist that we're going to turn you into a biologist, and if you're a biologist, I don't think we're going to turn you into a physicist over the next 2 weeks, but we want you to have enough of a vocabulary that you can sit down and have a conversation, that you can know that if your colleagues propose an experiment, you at least know enough to ask relevant questions about whether the science and the experimental design makes sense. So that's really, that's sort of the fundamental background of CEMB. So we think we're special not only because we are trying to combine the physical and the biological sciences, but also because we incorporate plants and animals, and I think that does make this center unique. I would encourage you, no matter what your background is and what your plans in graduate school are, to listen hard to the plant biology talks, if you're not a plant biologist, to listen to the mechanobiology of molecules talks, to listen to the introduction to modeling talk, no matter whether that's up your alley or not, and whether you can ever envision yourself thinking along those lines or not. Because one of the concepts is that, that we have is that there are fundamental mechanical properties that are shared by plants and animals. And if we can understand both plants and animals, we may get insight into mechanobiology in general. So this is the lectures that you're going to hear this week, and I'll tell you a little bit more about them in a minute. You don't get graded on them. They're all taped, so you don't have to take notes. I just encourage you to sit back and really think about what you're hearing, and think about how learning about what you learn about basic plant biology might be relevant to your work in materials science, for example, or how what you hear about modeling might be relevant to some of your basic cell biology work. Just try to let your mind make connections, if you will. CEMB has really 5 tiers of educational programs. So first is we have, our goal is to start training undergraduates through an REU program, that's research experience for undergraduates. At Penn, we have 10 undergraduates here this summer from a variety of schools. And can I just ask you guys to stand up? I'm sorry, I know that's goofy, but yeah, and if you could just go one by one and just say your name and the school that you're from. That's all. You want to start here? John?
I'm John Durrell from the University of Virginia.
I'm Brianna
I'm from Washington.
I'm Christine Mister. I go to Cornell. Olivia Levitt from Worcester Polytech.
Kamani Driver from Lincoln University.
Victor Morales, University of Puerto Rico.
Cassie Law from our college.
New Jersey Institute of Technology.
Okay, thanks guys. So these are our undergraduates. That's a very important part of our program is training undergraduates. We have a great group, and I think you'll be encountering these students as you go through your lab rotations this week. We also have 5 students at Washington University in St. Louis. That's not a formal program yet, but those 5 have been interacting with our REU students. The second tier is that we have a goal of having trying to teach this kind of integrated education, integrated science education at the high school level. As many of you know, when you get to college, and when your colleagues get to college, people have often defined themselves as either biological or physical sciences before they even walk in the door, and we want to change that for at least a subset of students. Rather than design a teacher program ourselves, we've actually brought in this year 3 master teachers, highly experienced teachers with insight in curriculum design, who've participated in teacher research programs in the past. We brought those 3 in to design a program for us, and I just want to ask those guys to stand up. And if you could just introduce yourselves and what school you're from.
Sure. I'm Catherine Calla. I work for the School District of Philadelphia, and I teach at Crick Learning Center High School.
Hi, I'm Rich Staniak. I work at Upper Gilman, and previously thought so.
Good morning, Mark Hayden. I work in Central Box West in Doylestown, Pennsylvania.
Okay, thanks guys. So the 3 school districts that you heard about are in 3 different counties in the area. They serve a wide variety of students, and they're all very interested in this integrated science education. I'm going to encourage the teachers to talk to you if they have a chance in the next 2 weeks about what did and didn't work for you guys at the high school level. Now that you're in graduate school, what does your perspective tell you? Similarly, if you have things you really think they should know about high school science education and how it might be done in a more integrative way, you've seen them. Our third program is postdocs. We have a variety of programs for them to encourage collaborations, giving them the opportunities to travel to other sites. I should say CEMB is a 7-site center. Our sites include not only University of Pennsylvania and Washington University in St. Louis, which are the 2 largest programs. We also have Bryn Mawr College, which is just in Bryn Mawr outside of Philadelphia, New Jersey Institute of Technology, which is in Newark, University of Texas at Austin, Alabama State University and obviously Alabama, and Boston University. So postdocs, as well as advanced graduate students, have the opportunity to travel to other sites, to engage in short-term collaborations, to learn techniques, and so on. So keep this in mind as you continue your graduate program. Faculty are also are heavily involved in educational programs. We encourage them to take sabbaticals in each other's labs, and we have weekly working groups, and which I hope many of you have participated in already, where we talk about various aspects of mechanobiology and the work that's going on. So the most in-depth program is for our graduate students, and we're trying in a variety of ways to get you guys to integrate physical and biological sciences, giving you opportunities to collaborate and learn from other sites, as I just said, and there'll be fairly extensive innovation training as well as this boot camp. And the goal of this boot camp is to start to bring you guys together to form a peer group where you can hopefully learn from each other. I have heard from your mentors about what your backgrounds are in the last week, and I know some of you really have absolutely no biology experience whatsoever, and some really have no math or real hard science background since college. And so the goal is not to turn you into the same thing, but to get you to the point where you have some common ground for collaborations. This boot camp was organized by Ram Dixit, who's at WashU in St. Louis. He's going to give the second lecture today, as well as a number of colleagues. And the week 1 format, I think you've all gotten that in the mail from Jim, is we have morning lectures, roughly 3 hours of morning lectures. Most mornings that's just 2 lectures, and those are open to everyone everyone in the center and really anyone at the university who might be interested. Some of these are actually remote, and we apologize for that. We couldn't get every single person on site for this week. But our rationalization is that when you're carrying out collaborative science, in many cases it's through FaceTime or GoToMeeting or Skype, and this is just something to get used to. So apologies in advance, but that is hopefully it will at least be useful to get that experience. We also, for the graduate students, and this is limited to the graduate students and to the teachers, we have these midday enrichment seminars that are talking about team building, about careers. We have a variety of, we have some people from the Penn Center from Innovation to talk about technology training. Jess Teitel, who is at Boston University, she heads their Biological Design Center, is going to talk about doing good science and reproducibility, and that may seem like an obvious topic, but we both have a number of sort of horror stories about papers that were published in really stellar journals, and there were key controls that hadn't been done. So we want to try to give you a framework for working for avoiding that. And finally, each afternoon in the first week, we're going to have hands-on labs with postdoc TAs. These labs are limited to the graduate students in the program. And so what is going to happen with these labs is, let me just see. So today, we're dividing you up into two groups, and I'll tell you what those groups are in a minute. The more math people are going to go into a lab learning about basic biology, cell biology techniques, and the more bio people are going to do some basic math and modeling work. Other than today, you guys are going to stay together as a group. You'll have most afternoons 2 different kinds of labs, just to give you exposure to some basic techniques. Okay? The TAs who you're going to be working with are this group right here. I want to give a special shout out to Graham. Is it Graham here? Yeah, Graham. Graham is our TA from WashU. He's the person, because all of the plant work in the center is done at WashU, we don't have that expertise here at Penn. Graham has come. He is your point person for any plant-related issues in the next 2 weeks. And for half of you, that's actually going to be a big deal. So, as I'll tell you in a minute. And I think Jim can give you contact information for these TAs. Even though they'll be working with you in labs this week, they're available for the whole 2 weeks, and when you do your final project in the second week, they're available for you. So in day 1, you're going to either get tutoring in math and mechanics or in biology. I've divided the 2 groups as shown here. Ricardo, Aaron, and Ryan are going to go into the math and mechanics lab. Sajia, who's not here, is an advanced graduate student who is supported by CEMB, and she's going to help out with that particular lab. In the basic cell biology lab, and that's with Fareed and Schwan, the basic cell biology lab will be Gary and Graham, and people with that will be Richard, Andy, James, Tam, and then Dongning and Tanya are also going to help out with that lab. So there should be more than enough people to give you one-on-one help in both of these topics. And if you need more information, you can talk to me at the break. So week 2 is going to start on Monday morning with what we call an innovation slam. So innovation slams, these were designed by Rob Mock with Dan Ha and Chris Chen. These guys all have significant experience with commercialization of their ideas. And they're working with the Penn Center for Innovation to put on, there are going to be 3 sessions, 3 4-hour sessions over the year. The first one will be Monday, and then there will be another one in November, and the third one probably in the spring. And you're going to be, you will, during that period, actually work on a specific project that you may come up with, maybe not, come up with some ideas for commercialization for. You're going to hear more about that in the topic that you're assigned, which I actually want to be the advertising manager for because it's pretty cool. So that'll start Monday morning at 8:30. That's also going to be open to postdocs in the center, so it'll be more than just the graduate students. Okay. And then starting Monday afternoon and all day Tuesday, we have a whole center retreat. Everyone in the center, every postdoc, every undergraduate, every graduate student, all faculty members will be involved. We really want you to participate. There are going to be a number of small group sessions talking about how to make this collaborative science work, what kinds of projects, what big questions are we asking. This center will not work unless graduate students and postdocs really buy in and are involved. So we designed this retreat, Liz Haswell, who is in charge of it, designed this retreat so that everyone is involved. So your input is very definitely needed. So finally, the final project, what we're going to do is we're going to divide you into two groups, and I'm going to give you you're going to get two real scientific problems that are involved. In the second week, you're doing experiments and also carrying out some modeling. We're going to ask you to start thinking about and planning your work in week number 1, and particularly if you're going to need any reagents that you can see, please let us know so we can order them. And you're going to carry this out in week number 2. The TAs are all available for advice and logistics, as are all the CEMB faculty. And I can meet with each group after the talks today and give you some ideas on who can provide advice. But we have lined up people to give you specific advice. And then a week from Friday, you're going to give final presentations on the work that you did. These are problems we don't know the answer to. They may lead to some interesting science in the center. So before I tell you some details about what those two projects are, I just wanted, I want to have all the graduate students introduce themselves. So if I can have you guys, actually I'll have you stand up one at a time. How's that? And then you can sit down. So I'm just going to go in order according to my list here. So Richard, just say your name and where you're from.
Guys, I'm Richard Vincent. I'm a first-year PhD student from Nutrition Technology.
And he works with Bill Hunter and Trina Arnsay. You'll meet Bill at lunchtime today. Next is Ricardo. Is Ricardo not here? Okay. Dong Ning.
Hello everyone, I'm Dong Chen from Royal Lab in UK.
Aaron.
Hi everybody, I'm Erin and I'm a second year.
And who's on?
Both my staff and Erin.
Okay. And they're both of them in the center. Ryan.
I'm Ryan Calhoun. I'm from Ron Dixon's lab at Washington University in St. Louis.
Okay. Andy.
Andy Clark, second year graduate student from our college.
James.
Jake Meyer. I'm at WashU and I'm under Dr. Michael Slauson.
Okay. Tam.
Sam Talbot. I'm in Dr. Gary Dean's lab, Alabama State University.
And Tanya.
Hi, Tanya, second year in Rock here.
Okay. And Sajia, I think, is not here. So, all right, welcome you guys. So these are there are two topics. Topic number 1 is on pollen tube mechanics. If you get assigned to this and you don't know what pollen tubes are, have no fear. Really the idea is to teach you some concepts of mechanics and collaborative science. It doesn't matter that these are plants and not animals. So this is, we're going to be asking you to answer specific questions about whether or not substrate stiffness affects pollen tube germination and growth rate in vitro. Jim McGonigal has collected pollen tubes from lilies, so we actually have these, and we're gonna ask you to model what you observe. The second topic is on tissue mechanics, and the real question is what determines the multi-axial mechanical properties of a tissue. We're going to ask you to choose a tissue, and we'll give it to you, and you're going to do some mechanical measurements, and then try to model what actually determines the multi-axial mechanics of that tissue. So these are the groups. Sajia, Richard, Aaron, Tam, and Ryan are in the pollen tube project, and then tissue mechanics is Tanya, Ricardo, Dongning, James, and Andy. So I just want to tell you, if you feel that you're completely lost on this, you have no idea what a pollen tube actually is. We have arranged these groups so that it's a mix of people with very different kinds of expertise. I assure you, you might feel you know nothing about it, but there are other people who feel they know nothing about some of your topics. So the group should actually be balanced out. And as I said, we have plenty of support for you. There are some social opportunities. Tomorrow night, Vivek Shenoy, who's the director of the center, and I are offering anyone who wants to, we'll take you out to dinner. These are all completely optional. and any other faculty who want to come can come. On Wednesday, we're going to have a happy hour with the TAs and other non-faculty CMB members at the New Deck Tavern, and we'll give you details Wednesday morning. And then if we can find an additional faculty, Rick Asoyan, will take you out to dinner on Thursday. Okay, so we'll give you plenty of opportunities, I hope, to meet CEMB faculty, but we also hope that you have lots of time to talk to each other. So finally, these are the resources, and I can ask Jim to send this slide out so you can, you know how to contact him, you know how to contact me anytime. Ram Dixit at WashU will be here for the retreat, and you can reach him by email. Particularly, he's a real plant resource. Please, please, please let us know if there are any problems at all. We've sort of blocked off time the next 2 weeks, so we're available to come and deal with any issues. This is our first boot camp. I mean, CMB was just funded in November. We really want to get your feedback. We have Lizanne DeStefano at Georgia Tech is our evaluator. We contract with her to do evaluations. You guys have, I think, already heard from her. She will be contacting you again for feedback. Please fill out those surveys. It matters enormously to the NSF that we do that, but also informally, just, you know, please feel free to give me, Jim, Rick, anyone else feedback. So with that, I will introduce the first lecture, and that is Paul Janmey. Do you want to wait 3 minutes, Paul? Or?
We'll need to load up this.
Oh, okay. So,
why we're interested in mechanobiology? What's why kind of why now, and why is it useful to combine physics and biology? And I want to do that partly by telling you what the kind of the motivation that has driven people who work in this space, why this has become more popular, or possibly more productive. And that is that as people have started to measure how really simple things like how stiff are different tissues, you know from your own intuition that different tissues have different mechanical properties. And it turns out that if you measure that thing systematically, they're surprisingly well controlled. So tuning the stiffness of your liver seems to be a physiological thing, just like tuning the calcium concentration in your blood. If it goes awry, bad things happen. But even though we know so much about what happens when chemical concentrations change, we know much less about what happens systemically when the stiffness quantified by an elastic modulus. And how many of you know what an elastic modulus is? Whoa, okay, surprisingly many. Okay, so my hope is that everyone will know it, at least in an intuitive way, and there's no sense in making things too mathematical because we don't really have to. Okay, so tissues have this surprisingly well established stiffness, but cells don't. Cells, if you take a cell out of a tissue and culture it in a dish, that cell can change its own mechanical properties over a very large range. So it's not the cell that's sort of genetically destined to have a particular kind of mechanical feature. Its stiffness depends on what the environment is. And so that turns out to be kind of a useful thing to be able to manipulate. Because as people have done more and more looking at this, if you change the physical properties of a tissue in an animal, or if you change the physical properties of the material on which you culture cells in the lab, you could really change the phenotype and probably even the epigenomic kind of information in the cell just by applying physical stimuli. And it's actually not so surprising because all of our senses work on physical stimuli. They all change, sound waves or photons or something into a biological process. And the sense of touch, which is basically what cells are using when they manipulate the stiffness of their environment, that could also be translated into the same sort of signaling mechanisms that everything else can. And what's really driven this lately is that certain kinds of tumors and cancer, but certain other disease tissues, such as fibrotic tissues, they've been known forever to be stiffer than the surrounding tissue. And the interesting thing is that stiffness isn't just a cause of the disease, it might actually be contributing to it. And from a kind of a physical science point of view, it would be interesting to understand what are the principles that give the tissue the stiffness that it has. So if we knew it, we might be able to manipulate it. So I want to first show you a little bit of kind of background, a little history. It's sort of Penn-centric, but it turns out that in the 1920s, and I'm sure way before that, serious scientists at, self-identified serious places like Penn took this kind of study really seriously. And so here is one of the most influential physiology textbooks from the 50s or 40s, I guess, from this guy, Louis Heilbronn, who's in the zoology department here. And the preface of this book says, in any attempt to interpret the machinery of a living cell, it's essential to know something about the mechanical properties of the protoplasm, what we now call cytoplasm, in the cell that's being investigated. So that was kind of like perceived to be, yeah, everybody knows that, we should pay attention to it. And then in the advent of molecular biology, that went largely dormant, or at least took a sort of a back seat to the wonders that you could do with molecular biology and chemistry and genetics. So the other bit of history is also by coincidence from Penn. There's this really fantastic landmark paper from these two guys, William Tsai Fritz, who was at that point a postdoc or a grad student, who had traveled overland first to Switzerland and then to the lab of this friendly fellow, whose name is actually Freundlich, who is a physical chemist in Berlin. And what these guys did was they, especially him, they built the first kind of microscopic rheology machine. They did this in the botany department. So this guy is a botanist. And they built this electromagnet that they stuck the tip of it near a microscope slide, put this really nice thing together, and then they borrowed the micromanipulator from a neighboring lab in order to be able to shove a piece of magnetite into a really big cell. And the biggest cell they could find was some kind of plant cell. And they did probably the first measurements of intracellular rheology this way in 1923. And I think the movie won't play, and there's no time to show it, but presumably this will be recorded, so if you are at all interested, look up this YouTube video. It's just fantastic. It was made in the 1950s by the guy Tsai Fritz, who discusses how you measure mechanical properties of protoplasm and stuff. It's just a fantastic, weird thing. So this is also this idea that trying to understand the physical properties of cells might be useful was also the thesis work of this guy, Francis Crick, who's one of the DNA Crick guy. So that's not what he worked on as a graduate student. What he worked on as a graduate student was the physical properties of cytoplasm. And he basically took that method from that was done in Germany 30 years ago or so as his method to measure the viscosity this time in animal cells. And he made some measurements of how stiff the cell is. It's much softer than what we now think it is. But one of the interesting conclusions of measuring the stiffness of the cell is that even though he knew that the inside of a cell was more solid than it was liquid, so the cell is not just a bag of water where enzymes float, he did not like this idea that the cytoplasm, that the physical properties of the cytoplasm are any evidence for some kind of cytoskeleton. So that's basically the conclusion of this paper. But these days, most people that think about cell mechanics think about the cytoskeleton as dominating it. But that's not what he thought. He thought that was all nonsense. Okay, so here's the kind of experiments that maybe you'll measure later in one of the projects, or that have motivated a lot of stuff. This is what I told you before in kind of numerical form. If you quantify the stiffness of a tissue by an elastic modulus, and I promise you'll know what an elastic modulus is in a couple of slides from now. So this is measured in units of Pascal. So 10 Pascal is snot, and glass and plastic are gigapascal, and the kinds of tissues that most soft animals are made out of have elastic moduli of a few hundred Pascal to maybe a few tens of thousands of Pascal. So liver is a relatively soft organ, but when it becomes fibrotic, it's way stiffer. And when malignancy develops in some tissues, but not in all, but breast is a big example, as malignancy develops in the breast, it becomes much, much softer. And obviously this is why palpation or other physical probes are diagnostically useful. And the Asoyan lab has shown that even before you get sick, if you give a genetic mutation to an animal, in this case by knocking out ApoE in them, the aortas of these animals get significantly stiffer before they get sick. These animals are doomed to get atherosclerosis. But even before you see visible signs of badness happening in the aorta, it's already stiff. And that stiffness seems to contribute to the disease process. And I don't want to overstate the case that stiffness is a bad thing or the changes in stiffness are always pathological. It also just happens during normal development. So when a heart first develops and the first myocytes appear in the embryo, that little heart, that cardiac cushion where the first sarcomeres and everything is really, really soft. It's as soft as the bone marrow. It's really soft. But that same heart in the adult is orders of magnitude, like 1,000 times stiffer. And because the heart cells don't divide very frequently, some of the cells that were present here are also present there. They have just learned how to adapt to their new environment by expressing different proteins, different signaling pathways, and they've learned to cope. So you can cope with changes in stiffness, but if you don't, it's not a good thing. And here's an example from Becky's lab of what kind of, or how big a deal this can be. So if you manipulate substrates on which cells are cultured, in this case they're hepatic stellate cells from the liver, if you manipulate the stiffness of the material that you put these cells on, but don't do anything chemically to them, don't change any growth factors or other stimuli, you just culture them on things of different stiffness. If you take them from on stiffnesses that are about as soft as the liver itself, these cells maintain much of their kind of native phenotype. They store fat-soluble vitamins. They don't produce matrix. They're not particularly active or mobile. But as you increase the stiffness underneath them to create a mechanical environment that's like a fibrotic liver, that's enough to stimulate these guys to act pathologically, or at least differently. Okay. And I show you also this because actually, here's the kind of experiment that made that image I just showed you possible, and it's something you'll do too. I think increasingly people have moved from making in vitro, or literally in vitro cell biology by growing cells on glass or plastic in order to take nice pictures of them and immunostain them and whatnot. But because the phenotype of that cell is so different when you put it on something soft, you could also just put a soft cushion on top of these glass surfaces. The cushion is usually something like a polyacrylamide, the same thing you do electrophoresis with, but it could be almost anything that's soft. And then because cells typically don't like to stick by themselves to something like polyacrylamide, you covalently put some kind of protein that the cell will stick to. It can be anything, as long as the cell has an adhesion receptor, and then culture them on that. So that's where those hepatic stellate cells came from. And if you do the same trick with myocytes, with cardiac myocytes, this time taken out of a neonatal rat. So the rat's heart was about this stiff when the myocytes, when the heart got blown up to isolate the cells. So in the heart, the cells looked sort of like this: elongated things, big stripy sarcomeres, very elongated shape. But if you take the same cell prep and just put them on something soft, much softer than the heart from which this came, these cells can't restore this nice phenotype. And if you put them on something really stiff, like glass or gels that are much stiffer than the normal heart, but about as stiff as an injured fibrotic tissue in a heart, they can grow and spread, and they make nice actin structures, but those structures aren't what the cell is meant to do. So stiffness can be a fairly important control factor, and therefore it would be nice to measure it. And here are two ways of measuring it. And this is probably most of the ways that all the data I showed you, and maybe some of you will do these kinds of measurements, or you already are. But this is how stuff is typically measured. You either put a little centimeter-sized disc or kind of cone-shaped little sample in between two plates. One of them doesn't move, and the other one rotates. And by putting torque on the sample, you can calculate how much resistance there is to the torque. And doing things in this way, where you apply a force parallel to the surface that you're touching, in the plane of the surface that you're touching, that's a shear deformation, and you measure a so-called shear modulus. And these kinds of measurements deform the sample without changing their volume. They just put a little lateral movement on them. But more frequently, and especially for small things like cells or biopsy punches or something like that, you cannot do this kind of measurement. So instead, what you do is you poke it. You push into the surface of it, or you compress it, or sometimes you stretch it. And so if you deform the material by putting a force that's perpendicular to the face that you're deforming, not parallel to it, but perpendicular to it, then you apply either compression or indentation or extension or stretch, but you don't, instead of shear. And if you do that kind of measurement of putting a uniaxial stress and uniaxial deformation, and you measure the uniaxial stress or the resisting force, you measure the so-called Young's modulus. Young is a really interesting guy. He was an ophthalmologist who also did a lot of physics, but he's the guy who figured out how astigmatism worked. And I guess that's related to the stiffness of the eye in some way. But that's for some historical reason, this kind of modulus is called a Young's modulus. And it is different from this because, in principle, you can change the volume. If you compress something and squeeze water out, the volume will decrease. If you stretch something and allow water or air or something to come in, the volume can get bigger. And the volume change difference between this and that mean that whatever you're measuring here may or may not be simply related to what's measured here. And one of the kind of things that you may run into later is that the engineering literature and basically soft matter physics and a lot of theory is works on sort of continuum mechanics kinds of models where at small deformation where these two things are really easy to relate to each other. And the unfortunate thing is in a lot of biological materials are anything but continuous, right? They got fibers going through them, they got organelles, they got little blood vessels through them. They're much more complicated structures, and what you measure with this does not necessarily tell you what you'll measure with this. So that's kind of an aside. Okay, but what is it we're really measuring? So the science of measuring the mechanical response of materials to forces is called rheology, from rheos or flow, most famous from diarrhea or rheostats. So that's where the word comes from. It's just a made-up word from, I think, again, the 1920s. And the idea is you apply a force to the surface of a material and you calculate how much it deforms. And there's only really two variables. One is a dependent variable, the other is an independent variable. So either you apply a force and measure how much it moves, or you move something a certain amount and measure how much force it pulls back with. So those are the only two things that go into all these measurements. It's very much analogous to measuring the spring constant. You take a spring, you hang a weight from it, you look at how much that spring elongates. It's basically that with some geometry mixed in. Okay, so you apply a force to a certain area, and the bigger the area, the bigger the piece of tissue, the more force you need to deform it. So you need to normalize by the area to generate a variable called stress. Usually it's shown as sigma, lowercase sigma. So force divided by area is something called a stress. And stress has units of force over area, surprisingly. And in SI units, force over area, in SI units, that's a Newton over meter squared, which is a Pascal. That's where Pascal comes from, force over area. So Pascal is a Newton over square meter, which is an unhelpful unit for tiny things. So it's the same thing as I always get this wrong, piconewton per square micron. Okay, a Pascal is also a piconewton per square micron. Same as okay. So if that helps your intuition, that's good. Okay, so this part is typically what you do to the sample, and you ask how much did I deform it, and oddly enough, quantifying the deformation is much more complicated mathematically. It's not that if it was just a linear spring, you would measure how much longer the spring got. But because samples are irregularly shaped and the forces are deforming the thing in a kind of complicated way, strain is relatively subtle to calculate. But strain is the measure of deformation. It doesn't have any units. If you have a really simple thing, like a cube, and you apply a force over the area of the cube to generate a stress, so you apply a shear force on the cube, on the top surface of the cube, force over the dimension squared, and the little face of the cube changes from a square to a trapezoid. Not a trapezoid, a rhomboid. What do I mean? Rhombus. Is that right? Yeah, a rhombus. A parallelogram of some sort. In this case, a really simple case, the strain is simply the distance that it moved over the height. So that will quantify the strain. It's never so simple in real life. If you have a disc-shaped sample or if you have a weird sample, the strains are a little bit more complicated to measure, but you can more or less intuitively get an idea that if you have a sample and I move it a distance x, and I divide x by the dimension of the thing that I just deformed, that ratio of how much I deformed it to its height is more or less the strain, order of magnitude. So what it means is that like a 1% strain is a tiny deformation. 100% strain doubles the length of the sample. Okay? All right. So those are the two things that are important for determining the force. I mean the stiffness of a material. How much force you apply to the sample during a measurement, and how much deformation did that force over the area achieve. So stress over strain. If you divide stress by strain, you get the shear modulus. So here again, there are two ways to deform these things. You can either deform them the way I showed you now. You take a square face at the beginning and you deform it to something that looks like that. That's a shear deformation, and the strain is roughly speaking this movement away from the square divided by the height. Or you can apply the force perpendicular to the face and move it and stretch it out or compress it. That's actually a much more simple way to quantify strain. In this case, the strain when you stretch it is just the increased area, the extra area you put in, divided by the initial area. And there are again various nuances and different conventions for how to quantify that. But that usually is some variation of how you measure strain. And again, strain is dimensionless. So stress is force over area, or Pascal. So that means that if you define the elastic modulus as stress over strain, that thing also has a unit of Pascal. So that's a little bit, that can be confusing sometimes. Stress and elastic modulus have the same unit, but they're not the same thing. It's just that stress divided by strain, and because strain has no units, gives you modulus that is also Pascal. Okay? All right, so I will not go over this, but it's in the notes if you want to remind yourself of what everything is. But there are two bits of jargon that are really useful to get straight. So stress and strain are kind of what I just told you, and they're sort of what we mean in regular speech. But stress and strain could often be conflated. So strain, again, is just a geometric deformation. It might be really hard to do that, might be really easy to do it. It's just a geometry change. Stress is the amount of force that it took to do that. Okay, but there's also two other things that are really important for tissue mechanics, and that is to differentiate elasticity from viscosity. And this is, in literature, this is one of the things that I think that is kind of, hard to get over. Not hard. It makes access from a non-specialist more of a pain in the butt, because it's just so jargon-filled. And it's partly jargon-filled because what elasticity means for soft matter physics is not typically what we think of it intuitively. For something to be elastic doesn't mean just that it's stretchable. It doesn't mean soft. Something that's elastic is something that if I apply a force to it and the material deforms, if I let go of the force, the material comes back exactly to where it started from. That's what elasticity is. The ability to totally come back to the initial place after having been deformed. You could be rigid, it could be soft, right? The softest rubber band and a piece of steel, they're both perfectly elastic. It's just the one is soft and the other one's hard. So elasticity is not the same thing as soft or stiff. It just means can you recover all the force that you put into it? And another way of thinking of it, which is kind of a useful thing in many ways, is that when you deform a material, you're doing work to deform it, right? So that's an energy. So you've invested some energy pulling on something. If the material is elastic, all the work you did pulling it is stored in the material, so that when you let go, the material will give you all that work back. Okay, so that's different from what I'll tell you about later for viscosity. Is that more or less clear? And so one of the things that you'll see sometimes in papers and whatnot, you'll probably even hear it from some of the labs, is the storage modulus, the shear storage modulus. A storage modulus is the same thing as the elastic modulus. That part of the stress divided by strain that is completely stored in the material and is totally recovered when you let go of the force. The other thing is viscosity. And so one thing I've glossed over in defining the modulus and stress over strain is that in a real tissue, if you apply a constant, well, what's a good thing? What's a good, all right. Okay, think about, I can't think of anything less gross at the moment, but just think about a wad of spit, right, which is partly elastic, but it's also very viscous. If you have some spit dripping from your mouth, or let's say you have like a really bad cold, and so it's like really gooey, it's the force of gravity that's pulling it down. But the longer you sit there, the farther that thing will deform, right? So if you just measured stress over strain, so the stress is a force of gravity over the cross-sectional area of your spit, then that, and divide by the strain, and to give you an elastic modulus, that number is changing as time goes on. At the beginning, it's a fairly small number. But as the force is always the same, the force of gravity is always the same, but as time goes on, the strain gets bigger and bigger and bigger. So if you divide stress by strain, the elastic modulus goes down, or whatever that modulus is. So, and if you somehow became weightless, the piece of spit would recoil to some extent. Not completely, but partly. And so part of the stress is stored, and part of the work is not stored. And so you have to differentiate the elastic from the viscous part. So that's what viscosity is. It's a response to the force in which the material deforms, but in principle without limit. It can keep moving and moving and moving, and if you let go, it'll just sit there. So, and again, viscosity is not, thick or thin. Anything that has a time-dependent deformation in response to a constant force is viscous to some extent. And the viscosity is not proportional. Viscosity is a number that is not proportional to the stress. Well, it is proportional to the stress, but it is not proportional to the strain. It's proportional to the strain rate. So viscosity is the ratio of stress to the rate of strain, how fast the thing drips or how fast the thing continues to deform. And so strain rate strain doesn't have units, but strain rate does have units of reciprocal seconds. So viscosity has units of Pascal second, because it's stress divided by strain. Okay, and most things are viscoelastic, right? They have elements of both. All right, so here's why these stresses and whatnot are important, because they show up everywhere in biology. And I'll give you a couple of really quick examples. They are are wildly important in the vasculature because fluid shear stress, as it flows over the endothelial cells and other structures of the blood vessel, really changes the biology of the cells underneath it. And so if fluid flow changes or the resistance of the cells to that fluid flow changes, lots of things change physiologically. There's also all sorts of stretch and tension. Every time you breathe, every time your heart pumps, every time you sit down, you're putting all sorts of either circumferential tensions, and especially think about your aorta. Every time your heart beats, the beginning of the aorta balloons out and comes back. And you want it not to balloon out too much or things get bad. And so the tissues are designed in this really interesting, complicated way to prevent that. But these kinds of deformations, which are fairly big, they're strains of tens of %, happen all the time, and the tissues have to be able to deal with that. And I already mentioned this kind of substrate stiffness. As the tissues get fibrotic or diseased, things change, and there's a lot of compressive stresses. And so some of you probably know this thing called Wolff's Law, which was known from the 1800s, where the bones grow as a function of how much pressure is applied to them. If you stop, that's why astronauts and cosmonauts lose bone mass, because as you stop putting gravitational force on them, the bones remodel differently. And so these are very, very big forces. They're not very big strains on the bone, but the forces are enormous. The stresses are really big, and all these tissues accommodate themselves. I think I have time to show you this one quick example of the shear stress in pathophysiology. So this is an area of mechanobiology that is really classically, this is one area that people have worked on for decades. It's not kind of recently rediscovered. It's a really very strongly worked out thing. And so here's just one example. There's no movie with this. But if you compute by looking at the blood flow through this branched blood vessel, what are the shear stresses that the fluid flow exerts on the endothelial cells underneath it? It turns out the fluid shear stress, if you combine it or if you compare it to the amount of nitric oxide produced by nitric oxide synthase, you can really change the amount of activity of all sorts of enzymatic reactions and all kinds of cellular effects that respond very much to the level of shear stress caused by the flow. And if you disturb the flow or change it differently, cells react. And they react in this fairly dramatic way. That here's an example, actually from Peter Davies' lab, who's just underneath us in this building. If you culture endothelial cells, as probably many of you do, under static conditions, just in a culture dish, or if you take them out of a tissue, and where a blood flow has been occluded, and you stain their cytoskeletal elements, their microtubules, or their intermediate filaments, or you just look at the shapes of the cells, they're these kind of cobblestone-ish sort of things with no particular directionality. Now, if you put laminar flow on the dish, it's not even a blood vessel, you just flow the medium across it, very quickly these cells reorganize to have a long axis and a short axis that points in the flow direction. And if you give the cell not sort of gentle laminar flow, but kind of turbulent flow, as would happen in a branched blood vessel, something intermediate happens. The cells kind of align, sort of, they don't know what to do, and there's a lot of disorder, and there's a lot of both genetic and biochemical signaling going on, some of which is clearly relevant to disease. I'm going to skip over the stereocilia thing because it's just so pretty. But I want to end with something that might be useful to later modeling is people's attempts to understand where this comes from. So I show you this picture just to kind of remind us that in real tissue, cells are always living in the soft world. So they have their own cytoskeletons made out of their polymers. But here are fibroblasts crawling through a collagen network to repair a wound in the lens, I think. And so their own natural environment isn't a cover slip or something like that. It's this three-dimensional, relatively soft matrix. And what are these matrices made out of? They're all made out of semi-flexible polymers. So both the cell interior, so here's the cortical actin network just underneath the cell membrane of a leukocyte, And here's this picture I just showed you of the extracellular matrix. All of these things, the collagen fibers, the fibrin networks in a blood clot, the cytoskeletal polymers, they're all made out of this class of semi-flexible polymers. They can make these beautiful interdigitated networks that connect one cell to another in an epithelial sheet. This is the aorta I mentioned. It's also made out of this complicated network of collagen and elastin fibers. But all of these things have a physical feature that they are made out of relatively straight, slightly flexible but relatively straight polymers, so that when you stretch them, the material can deform, and as long as the fibers are cross-linked to each other, what will happen is the shape of the fiber will change. It'll get elongated or compressed or something, but they won't let go of each other. So when you relax it, they'll pop back. And so this balance between elastic response and viscous response depends a great deal on how these cross-links are put in and how strong the fibers are to breaking and that sort of thing. It's that kind of stuff that sets the balance between the viscous and the elastic part. And the other thing that sets this balance is the way that cells are connected to each other or connected to the matrix. So here's just a cartoon from Albert's textbook, that there's a whole slew of different kinds of proteins that cadherins, but also many other transmembrane proteins that link one cell to another and that link cells to the matrix underneath them. So probably a lot of you work on this kind of stuff, but this is a key way that the cell is able to do that, to interrogate it. And so one of the kind of cartoon pictures that is relevant to understanding how cells might be able to sense force is that in the real tissue, they're connected. If here's just the end boundary of the cell, and here's the interior of the cell. There's an actin filament, a myosin head. The actin filaments are never attached directly to the cell membrane. They're attached through a whole bunch of proteins in series to a little focal adhesion place. The focal adhesion spot is usually mediated by a transmembrane protein that could be linked to this extracellular world. There's a million ways this can happen. But the idea is that if a cell pulls on this contact, as it does, and inevitably if a cell attaches to something, it almost always pulls on that thing. And if it pulls, it's pulling a bunch of molecules in series. And people, I think it's accurate to say that nobody really knows much of anything about how the cell does this or how the cell is measuring anything. But presumably its ability to measure things is because it's putting force, it's putting tension on a whole bunch of molecules in series, and depending on how those molecules unfold or interact with each other or break, those reactions can give rise to chemical cues that are just the same as anything else. I think I should probably stop here, right, to keep on time. So there's some other stuff in the PDF or the PowerPoint on kind of the physics of polymers of this sort. The idea is not for you to have to learn the math of how to understand the force extension relations, but just give you an idea of where this kind of mechanical response might come from. Okay. I guess, do we have time for questions, I guess? Yeah. Yes?
You mentioned turbulent flow in blood vessels. Is there any tissue failure that happens as a cause of turbulent flow in blood vessels?
Yeah, so Rick would be the guy to know this. So it's not so much, where the turbulent flow happens, which is typically at a branch or at a place where there's something bulging into the circulation, those areas of disturbed flow are prone to becoming atherosclerotic, or they're prone to becoming pro-inflammatory. As far as I know, there's no case, the forces that the cells experience underneath it, those forces aren't really big. They are not big enough to damage the cell itself. But they are, it's not so much their magnitude, but the fact that they're changing with time or they're changing with the direction, elicits cellular responses that are different from what it would do if there's just laminar flow. the forces that are, it's a really confusing thing, because it may be surprising, because if you've ever seen a movie of how fast the blood flows through a vessel, it flows enormously quickly. But the fluid shear stress on the top of the endothelial cell is actually very small. It's a few Pascal. It's very, very little. But the cells are still able to respond to it. And it's not super clear how they do it, but they're able to respond to it. But superimposed on that, and something related to things like high blood pressure, is the pressure in those vessels causes them to bulge out. And every time you have a pressure change, the blood vessels go in and out. So every time the heart beats, the aorta goes, like this. And so those pressures are huge. They are tens or hundreds of thousands of Pascal. They're big. They are really big. They're like, I think they're like tens of thousands of Pascal. So that's a lot to stretch these things. And so if you have defects in the collagen network that I showed you in the aorta, you can get the aorta break, or you can get a blood vessel to break in your brain because the pressure changes just get that thing to rip. So it's not so much the turbulence or non-turbulence; it's the pressure that's the destabilizing thing. Other questions? Okay, so hopefully we're on time.